Buenos días a todos y todas. Les damos la bienvenida a este nuevo webinar sobre COVID-19. En esta oportunidad vamos a contar con un disertante internacional, con el doctor Andrea Cossarizza, a quien le agradecemos porque sabemos que está sumamente ocupado y para nosotros es un placer poder contar con su presencia. Así que le voy a dar lugar al doctor Matías Ostrowski, que va a coordinar la presentación del doctor Cossarizza. Muchas gracias a todos y todas.
Gracias, Analia. So I will switch to English. It's really my pleasure to introduce Dr. Cossarizza. Dr. Cossarizza completed his MD degree at University of Padova in Italy, and then he did a PhD in oncology from the University of Modena and Reggio, Emilia, and the University of Bologna, also in Italy. After specializing in clinical pathology, he obtained an associate professorship at the University of Modena and Reggio Emilia, and in 2010, he became a full professor in pathology and immunology. At present, he's Vice Dean of the School of Medicine and Director of the School of Clinical Pathology at the same university. In 2016, he was elected President of the International Society for the Advancement of Cytometry, and his primary research focused in identifying the molecular and cellular basis for the involvement of the immune system in disease and infections, including HIV, AIDS, and sepsis, as well as its role in the pathophysiological conditions related to aging and neurodegeneration. Dr. Cossarizza has notable experience in the development and use of new flow cytometry approaches in immunological research. He has published more than 330 papers in peer-reviewed journals and is a member of several editorial boards of international journals. From the first moments of the pandemic, of the COVID-19 pandemic, he has been studying immunological changes in COVID-19 patients and the efficacy of novel immune therapies. So Dr. Cossarizza, we're happy to have you here in this webinar, and thank you for sharing your talk.
Muchas gracias. Thank you very much, Matthias, and thank you, Emilia, for the kind invitation. It is a big pleasure to be here in Buenos Aires, maybe, and to share with you the some data that we have obtained in the last months in my lab. So this is the cathedral in Modena. It's a very nice monument built in 1099. So it's just to start with a nice picture. And first of all, I have to disclose my interests, and we have received in the last months grants or help from a number of people and a number of companies, including Lengas, Bank, San Felice, Bank, Biper, Gianni Givellini, and then companies that are involved in biotechnologies and in biomedical research and development, like Biotechne, Thermo Fisher, Beckman Coulter, Beckman Dickinson, Aginet, BioRev, just to mention a few, and then a lot of people who gave us a big support to perform the studies that we have done here. So my talk will regard a little bit of what we know, and especially what we knew at the very beginning of the pandemic. I will mention how we have been studying T cells. I will talk about the cytokine storm and the production of cytokines in vitro, B lymphocytes and monocytes. These are the last studies that we have performed, and finally some conclusion. So what happened? I tell you a story. The story was that in January, it was very clear that something horrible was happening, would arrive very soon in Europe and all over the world. We saw the first patient in Modena at the end of February, and the first patient were seen in it 1 week or 2 weeks before, but it was clear already in January what was going to happen. And so I prepared my lab, and I organized the lab in different parts, in a part in which we could receive material from patients, I mean blood essentially, in a part in which we had to work with blood and cells, and a part for the molecular biology, so to divide and be safe working in the best condition, in the best possible conditions. At that time, there were a lot of questions, especially because Europe was not yet infected. I mean, the infection arrived in Europe in February, more or less, but the first patients were seen in Italy. So unfortunately, we were the first to experience the pandemic, and it was an immediate problem for us to understand what we had to do. So we start at the end of February, as I mentioned, and we had these questions that I could write and publish in cytometry in March, after 2 or 3 weeks, and I was very fast in preparing a short note or a short paper that was published, and now it's one of the most quoted paper, the most cited and downloaded paper ever. And because we simply were seeing patients, but we did not know why many of them had a very severe disease and died, while others remained asymptomatic or had a very mild infection. We did not know the mechanism by which young children were spared from a severe disease. We did not know the role of the immune system in the pathogenesis of the COVID-19, especially in the severe forms. It was not clear what was happening if we had immunological markers predictive of the severe disease and poor outcome, and we did not know whether there were markers associated with a good response to treatment, even if I have to tell you that in that period there was no treatment at all. So there were nothing to do with patients if not support them with oxygen and cortisol or whatever, and steroids. And so these were the problems that we had in the very beginning of the pandemic, and I wrote this again in cytometry. Then we started to see patients and to study a little bit in detail what was happening, and we could analyze, first of all, 21 patients who were admitted in the infectious disease department in the university hospital over here in Modena, and we sent the first paper at the end of March, a paper with a lot of data on these patients. The paper was received by Nature Communication, and then the reviewers were able to give us some very nice inputs to improve our study. So we had to enroll another 18 patients, and all of these patients had severe pneumonia induced by SARS-CoV-2, and the symptoms were classical symptoms with sore throat, fever, cold, dyspnea, chest pain. SARS-2 was detected with the nasopharyngeal swab, and all patients had pneumonia confirmed by X-rays. There was no need to perform a CT scan because it was absolutely easy to perform the diagnosis. What we saw, first of all, was that lymphocyte count was low, and in fact, the patients had a median value of 1,076 cells per microliter, less than 1/2 of the normal values. I have to say that it is not a trivial data, because in that moment, the data were not present in the literature. I mean, we did not know what was happening. There were just a number of reports by Chinese scientists, and in January or February, very little data were available, and most of them were not published, were public. I mean, because you could download the submitted manuscripts, but there were no paper that had peer review, and I would say paper that were very reliable. So the first problem was to understand what was going on in the field of CD4 and CD8, and we started to analyze in depth the subpopulations of CD4 and CD8. We could do this because, honestly, we were quite fast in finding all the reagents that we needed. And this was because in January, when it was clear what was going on, we could order a number of antibodies, kits, reagents, and so on, to be ready to start immediately. And in fact, this was what happened to us. So what we found was that the CD4 cell count were lower, much lower, even if the percentage of CD4 cells were more or less similar to healthy donors. And the percentage was similar, but the absolute number was dramatically lower because of the lymphopenia. And it is still not clear why people have lymphopenia in such a way. And then what we have done the first kind of analysis. We perform a 16-color flow cytometry panel with collaboration with Beckman Coulter. We could detect a number of these different cells. And the first analysis was performed by the classical gating strategy, in which you perform a gate on physical parameters and time, then a gate on CD3, on CD4, you identify CD4 cells, then you can identify cells that had markers of activation, synergia, exhaustion, differentiation, or Tregs by using, for example, CD38, HLA-DR, CD57, PD1, CD45 arrays, CL7, then CD25, CD127. Then within all these possible populations, we detect other cell types, like, for example, T stem cell memory that were identified as cells positive to 4 markers that are CD45 array, CL7, CD27, CD28, and CD95. And of course, this is the classical fast antigen. Then we could detect different types of Tregs, naive, central memory, effector memory, or terminally differentiated. Then, with this classical approach, with the classical manual gating, we identify main T cell populations, naive, central memory, effector memory, or terminally differentiated cells, and these are the percentages that were not dramatically different from controls, while the absolute numbers were lower because, again, of the lymphopenia. And this was very, at this point, this is what we expected to see. Then we found that patients had a high amount of activated cells, but also of cells that had markers of senescence, like PD1 and CD57. They also had a little bit more of Tregs in terms of percentages, while the number of cells was not statistically significant in comparison with controls. Then we perform another type of analysis, which is very nice, at least for the colors, and we perform the unsupervised classification of CD4 T cells with flow sum and with the uniform manifold approximation and projection system, which allows you to compress the data and to classify in 2 dimensions a number of different populations. As you can see here on the right, you can identify naive central memory, naive T stem cell memories, and naive cells expressing CD38, naive expressing CD-DR, central memory, and all the other populations. So in this way, if you are a little bit able to use unsupervised methods like TSNEO, WISNEO, or this one, you which works under R, you can easily perform the heat map and recognize different populations here as the expression of different cells in controls or in patients with COVID, with COVID, and the comparison between the 2 groups showed that there were dramatic differences in some CD4 T cell populations, as we found with the classical gating strategy. So it was nice to see that the results were more or less the same, and patients had dramatic changes in Tregs, in effector memory, in central memory, and in other T cell populations. Then we had some comments from the reviewers, and they asked if we could better characterize CD4 cells. So what we have done was to set up a panel, and this was done by Sara De Biasi, who gave a webinar a couple of weeks ago, if I remember well. And Sara was able to set up a protocol with 12 colors in the moment of the maximum lockdown, and we were able to get the reagents. I don't know how, but they sent us the reagents to detect simultaneously the presence of mass regulating gene expression like Tibet, GATA3, or and associated with molecules that recognize markers on the cell surface, like different chemokine receptors like CCL3, CCL4, CCL6, and CD161. And you can see here on the top there is a patient on the top panels, and the lower panel controls. And what happens? Again, we could see that the percentage of cells that express some chemokine receptors were different between patients and control. And for example, they had a low amount of CCL6 and a high amount of CCL4. So it means that probably there are some consistent changes in the capability of CD4 cells to home to different parts of the organism, different tissue organs. Then another interesting story was to combine these markers and to see that, for example, patients had a very low amount of cells that were able to co-express simultaneously CD161 and CCL6. And typically, this is TH17 cells, even if we could not detect raw gamma T because of the lack of the antibody. It was our problem, I mean, not to have all the reagents we needed. Then what we studied, we took a look to T cell proliferation, and with the method of CFSE and the cell trace, and what came out is that to make a long story short, you can detect in the same moment the proliferation index and the percentage of cells that could divide. So it means how many cells enter the cell cycle and how many cell cycles the cells were able to perform. So what came out is that very little changes occurred in patients in comparison with controls. That it means that if you can take the cells out from the organism and stimulate them, they work pretty well. Not only they work well, but also they have a normal functionality in mitochondria. And in fact, thanks to the help of Agilent that provided us the seahorse machine in a few weeks, we could sort CD4 cells and analyze the oxygen consumption by these cells with the classical oximetry system. We performed the analysis in micro-well with a few 100,000 cells per well, and we could detect the different parameters like the basal respiration of mitochondria, the proton leak and ATP production, the maximal respiration, and the non-mitochondrial respiration in terms of course, oxygen consumption. And this is possible thanks to the possibility to use different molecules like oligomycin, FCCP, rotenome, and antimycin A that block or stimulate different enzymes of the mitochondrial respiratory chain. Then what happened? Actually, close to nothing, because controls and patients were able to work in the same way. So CD4 T cells from patients and from controls were able to use or consume oxygen in the same rate, indicating that mitochondria work very well. So CD4 T cells have very good mitochondria that are functionally active. Keep this in mind, because this will not happen for another cell type that is CD4 positive, even if at low density, that are monocytes. And I will show you in a moment the different behavior of monocytes in comparison with lymphocytes. Then we took a look to another group of cells that are CD8 cells, and we could detect them with the same approach. Similar story, percentage of the cell was similar to controls, while the absolute number was much lower because of the lymphopenia. Again, performing the classical approach, we studied the different subsets. But first of all, I want to show you that what we found in terms of CD3, CD4 cell count, and so on, was similar to what has been published a few weeks later by a group which is in China and in Italy as well. And I am happy to see that the same results that we found were found by other people in the other part of the world, I mean in China. Going back to CD8, we performed the classical gain strategy to analyze cells in different differentiated state, activation, synaptic exhaustion, T stem cell memory, and so on. And what came out is that there were consistent changes either in the percentage or in the absolute number of naive cells and central memory cells, which is not unexpected because it is a viral infection. I mean, SARS-CoV-2 is a virus, so it is very clear that the immune system can respond to a virus in this way. I mean, activating cells, and indeed, if you take a look at this activation of CD8, you can see that there is a high amount of cells in terms of percentage and of absolute number that have a huge markers of T cell activation. So the story is that you lose many cells and you acquire activated cells as far as CD8 T cells are concerned. There is also a little increase in the percentage of exhausted cells, but not in the absolute number. Then going back to the unsupervised classification of CD8 cells, what came out, again, you can recognize all the different clusters here, naive, central memory, transitional memory, and so on. And using a UMAP analysis, the heat map story, what you see is that many populations of CD8, more than populations of CD4, were consistently different between healthy controls and patients, and in some cases the p-value was of the order of 10 to the -8, 10 to the -7. And you can see here what happened in different CD8 cell populations. Interesting enough, the group in China did the same kind of studies, and what they found was that the degree of activation was directly correlated to the degree, to the severity of the infection. So they just measure the amount of cells of activation with HLA-DR and CD38, like we have done. Then they also studied PD-1 and they also studied TIM3. TIM3 is another marker of exhaustion, and the data are absolutely similar to ours. Then we took a look to the expression of mast regulator genes and chemokine receptors, also among the CD8 cells, and what came out is that we found an interesting population of cells like that recognized by CD161 and CD6, which was almost disappeared in patients in comparison with healthy controls. These are healthy controls, these are patients. Also from the statistical point of view, the difference was huge, and in fact, if you see to the bottom right of the graph of the slide here, that patients with COVID have a very, very low number of these cells that are double positive for CD6 and CD161, like we have found for CD4 cells. Well, it was well described that in a monkey model, in a simian model of a viral infection, where when infection goes to the lung, it has been shown that these cells disappear from blood and migrate to the lung, where they can produce IL-17 and they can recruit neutrophils. So there is a direct correlation between the lack of these cells and the gravity of the infection. And this is very interesting because we have found something similar also in the SARS-CoV-2 infection, not only in, so the phenomenon is real, it is not only present in monkeys or in simian models, but it is something that you could use to detect the gravity of the disease, I mean, to check for the presence of Th17, I mean, CD8 cells able to produce Th17. And I will show you in a moment why they do produce IL-17. Okay, what about T cell proliferation? T cell proliferation among CD8 cells was not different in controls in comparison with patients. And I have to say that in some cases, if we take a look to the terminally differentiated cells, they had less cells entering the cell cycle, but the number of cells able to divide was higher. So you see here the proliferation index is lower, while the percentage of divided cells is higher. But the numbers are not so dramatic, so I do not think that there is a significant role for these sort of cells in the pathogenesis of COVID-19. Another study that we are going to finish now has been done in collaboration with a group of Pia Kvisborg and Tom Schumacher in Amsterdam in a cancer center. And this is a preliminary report which is online. And what we studied was the T cell response towards peptides from ORF1AB, I mean from the operative frame 1AB polyprotein in patients with COVID-19. We studied 20 patients and we could analyze up to 500 predicted MHC class 1 epitopes. And we saw that, first of all, the reactivity of T cells were very common. And not only, but we could identify different T cell responses. And what came out is that in patients with different HLA-A antigen or class 1 antigen, it come out that this peptide, TTD and so on, is recognized by a huge amount of lymphocytes. You see here the responsiveness of CD8 cells to this peptide is of the order of 30%. Then we have done an extensive analysis of peptides like this in patients with different forms of disease, and we found that this was present in patients with either critical or severe disease and with different HLA. And so you see here the reactivity to ORF1AB from different patients. Then we could analyze the transcriptome of these cells, and what PIA was able to do was to perform single-cell RNA-seq with the Illumina system, and what came out is that you can recognize different clusters of cells are able to express different genes like IL-7 receptor, MK1, MKI67, and the granzyme B, and many, many others. And what we found is that some of the genes were upregulated in cells that were sorted by the capability to respond to the TTD peptide in comparison with control. And these are all molecules expressed in a different way in cells. In specific T cells are recognizing this peptide in comparison with those recognizing other peptides. And finally, we found that cells that were sorted for the responsiveness to the TTD peptide had a huge number of genes that were upregulated and or downregulated. You see here the list, the long list of genes that are related to cell homing and proliferation as well. So what was the take-home message of this part? The paper is actually in revision. We hope that the reviewers will like our changes. And what we found is that a number of CD8 T-cell responses were directed towards the operative frame 1AB polyprotein, and these responses frequently had a very high magnitude. So the comment is that since most vaccines now are directed against the spike protein, we have to take into consideration the possibility to add another target to the vaccine, I mean to the available vaccines, by using the ORF operative frame portion of the virus, because probably this would potentiate the activity of a vaccine against another part of the virus. And you know well that as immunologists, and my impression is that if you use more parts of a single virus, probably the response that you obtain will be much higher. But this is something that we have to evaluate with time. Second aspect that we were interested in was the cytokine storm and the production of cytokines. So in the first paper on March 19, we asked some questions of what we have to do. Then after a few weeks, the problem was how we can do this. So how means how we can handle and process blood from patients with COVID-19 to perform safely studies on T-cell phenotype and cytokine storm. So I wrote a paper that contained sort of guidelines for the analysis of cytokines in patients in plasma and to manipulate, to work with blood from patients. Actually, I have to remind you that there were nothing in the literature. I mean, we had no idea what this virus could do. We had information about SARS, the old one, but not so many. We had more information on MERS, the coronavirus that affects the dromedary camel and then humans. But there were no information about SARS-CoV-2. So I wrote this and actually to explain people what they have to do. So the first data we obtained on the cytokines were performed with a panel of 31 cytokines. Now we have developed a second panel with 62 molecules, which are still under investigation. I mean, we are still analyzing the data. But what we have done with the first panel was to see that the number of inflammatory molecules were much increased as expected. And what we found was that not only inflammatory molecules like TNF-alpha or interleukin-1 alpha or IL-1 beta or IL-6, which I will mention in a moment, were significantly increased in patients in comparison to control, but also molecules that are able to exert an anti-inflammatory action, like, for example, IL-10 and IL-13 and IL-4 were decreased. Well, sorry, were highly expressed, were increased in patients with COVID-19. So there was a sort of contrasting production of inflammatory and anti-inflammatory molecules in these patients. And again, these data were confirmed by others. And here you see that the group of people in China and in Rome had the same result as ours, not only for IL-6, but also for a number of other molecules, including TNF-alpha, IL-10, and many others, interleukins-1, and so on. So then what happened? There were already reports on the importance of IL-6 in the development of the pathogenesis of COVID-19. So this is just an example of a review of a friend of ours who designed what happened in the patients that could take drug able to control IL-6 activity. And what we did actually was to study immediately the possibility to treat patients with tocilizumab. Tocilizumab is a drug that can block IL-6 receptor, and in our hands, the results were those you can see on the right. If you study these are the cumulative probability of mechanical ventilation and death, over here on the top and on the bottom, the cumulative probability of death of patients. We performed a study on about 400 people, and what came out is that we could reduce the probability of mechanical ventilation and death of about 70% and the probability of die of about 80%. So it means that if you use this drug in the right moment, not too early because it is fully unuseful, not too late because it is unuseful, but you have a window of opportunity in which you can treat patients with tocilizumab and rescue their life. Again, to obtain a reduction of mortality over 80% is something absolutely incredible. We have not done a randomized control trial simply because we have seen what was happening in patient number 3. I mean, the third, after 3 patients in which the result was spectacular. I mean, these were people who were not breathing at all. And after 24 hours after from the first injection, usually the therapy is made by 2 injections, 12 hours, one after the other. And 12 hours later, after the second injection, patients start to breathe with their lungs. So it was really something impressive. And so we decided not to perform a randomized controlled trial, which is now ongoing in France, and the results are absolutely excellent. They are seeing a reduction of mortality of the order of 40–50%, even more. So cytokine storm in plasma was paralleled by cytokine storm in vitro, because we have analyzed the capability of T cells to produce different cytokines after in vitro stimulation. And what we have done was to detect the presence of up to 6 different molecules or functions in the same cell. With a multiparametric flow cytometry, we could detect the presence of TNF, interferon gamma, IL-17, interleukin-2, granzyme B, and CD107A as a marker of degranulation, both in CD4 and in CD8 cells. And what came out is that patients were able to produce much more cytokine in vitro than controls. I mean, they had a higher amount of cells able to produce different molecules, including IL-17, which is crucial to trigger the activation of neutrophils and monocytes, especially neutrophils and especially in blood. So we put out the hypothesis that these cells could be a target, or eventually IL-17 could be another target for intervention for treating patients, since there are antibodies like secukinumab that is able to block interleukin-17. And what was interesting is to see that the production of IL-17 was done not only by CD4 cells, I mean, that could be considered Th17 cells, but also from CD8 cells. Also, these cells were able to produce high amounts of IL-17. Then we checked with the method developed by Mario Röderer at NIH, the SPICE software. We could have detected the simultaneous presence of different cytokines, different functions in a single cell. And what we found was that also in the case of, also using this kind of analysis, the amount of cells able to produce simultaneously IL-2 and IL-17 was much higher in COVID patients than in control. In any case, COVID patients were able to produce, had a high amount of cells able to perform more than one function simultaneously. Then what about B cells? We developed another panel to study different populations of B cells, and you can see here the presence of memory switched, naive, transitional cells, memory unswitched, memory IgM only, plasmablasts that were IgM positive or negative, B cells, CD24 that were unswitched, and finally cells that had the without antibodies on the cell surface. And again, we use the same approach, the unsupervised approach, allow us to show that almost all B cell populations were changed. And this is reasonable considering the importance of IL-6 in the maturation and development of B cells. Actually, these data were published in September by Sarah, my collaborator in the European Immunology. The others were published in July in Nature Communications. So these are the clusters of B cells, and you can see on the right that almost all these populations were completely different in COVID patients in comparison to control. And you see here again the percentages and absolute numbers. The number of B cells is lower, the number of naive B cells is lower, and over here you see the number of memory and the number of percentages of memory switched and memory unswitched B cells that were much lower in terms of percentage on the left and absolute number on the right. This was true for both unswitched and switched memory B cells. But what was very striking to us was to see that plasmablasts were much higher, a significant increase in blood from patients in comparison with controls. And you can see here the percentage and absolute number of plasmablasts almost absent in healthy control and much higher in patients with COVID infection. So what came out? We applied another type of analysis, which is the CRISPR component analysis, to understand the role and the weight of different parameters that we could detect. So we took two groups of patients, those who died on the left and those who survived on the right, and we divided the CRISPR component space in two parts, one here in blue, where that represent people who died, and one in yellow represent people who were discharged. So, and these are all the parameters that are important in determining the survival or the death. And you can see here that the B cell parameters, like the memory, the population of memory cells or switch cells, or memory IgM only, or transitional, or naive, or the total B cells, were important in determining the survival or the death. It is not that this has to be increased or decreased. This is just the weight of this parameter that can go up or down depending on the situation, but this parameter has a very good weight, a very strong weight in the correlation of death and survival. And of course, parameters like the oxygen present in the blood, the soluble oxygen, or the P/F ratio are very important on the one side. On the other side, the SOFA score, the M parameters linked to inflammation like D-dimer or C-reactive protein are very important in the group of people who died. So this means that you can, in a way, understand what is actually happening in patients by controlling more than one parameter. For example, if you do only SOFA score, you have partial information. If you put together SOFA score and other parameters, you have much more information. The last part of the talk, last minutes, are devoted to the analysis of monocytes. And this paper went online 1 month ago, and it is published in EMBO, Molecular Medicine. And we are describing the bioenergetics and mitochondrial expansion in monocytes of patients with pneumonia. So first of all, we saw that monocytes have a change in their distribution. You know, there are different monocyte populations. And what we found was that in our patients, there was a decrease of classical monocytes and an increase of intermediate monocytes. This was true also considering the clustering, I mean the UMAP analysis. You can see and you can detect the classical, non-classical, intermediate monocytes that were able to express CSR2 or not. And what came out, sorry for this is a wrong color, and the intermediate monocyte were changed, but the classical cells, the red one over here, those indicated by the arrow, were significantly different also in the unsupervised analysis. You see here the difference is very significant. So changes in the phenotype of peripheral blood monocytes. Then other parameters were those relating to, for example, PD-1 expression, PD-1 and PD-L1. And it was very striking, very strong for us to see that PD-1 was expressed by some cells, not so many, but PD-L1, the ligand to PD-1, was expressed by a number of monocytes. You see here that classical monocyte had about 20% or let's say 15% of these cells were positive to PD-L1, while 50% of intermediate and 20% of non-classical do express PD-L1. This is just to representative example of a control and patient. And even if you check the median fluorescence intensity, you see consistent changes in the amount of PD-L1. So it means that not only there are more cells positive to PD-L1, but you have more expression of PD-L1 at the single cell level. And this is what the graphic shows you. But I was talking about mitochondria. What happened to mitochondria? Well, we measure with the system of the seahorse, the oximetry, the consumption of oxygen and the production of ATP, oxygen, free radicals, and so on. And what came out is that you see here the difference between controls and patients. The patients do show a decreased oxygen consumption rate, a decreased maximal respiration, protonic, oxidative burst as well, and a decreased basal ECAR, which is the acidification of the media, which is a parameter due to link to glycolysis, not to oxidative phosphorylation. So what came out, to make a long story short, is that the respiratory capacity, I mean the ability to use oxygen, was much lower in patients in comparison to control, as well as the oxidative burst. The oxidative burst is the capability of cells to produce oxygen-free radicals after the stimulus. And you see a dramatic decrease on this parameter, meaning that monocytes are not able to perform their function as they should do, because they do not produce oxygen radicals that are absolutely useful if a monocyte on the macrophage phagocytize something. This has to be killed. So to lose this function means that they have a decreased cytotoxic capability for eventually phagocytized bacteria. But you know, I like to see things and not only to see numbers. And so I asked my friends here in the department to take a look to the monocytes. And in the middle of over here, you see a normal cell. I mean, a cell from a normal from a donor, and these are normal mitochondria with a very nice mixture, very nice shape. with a small enlargement. But if you take a look to mitochondria from patients with a higher enlargement, you see how mitochondria look like and how they change, they shape, they have a lot of vacuoles inside, they lost the crispy over here, and so they are definitely dysfunctional. And if you measure a number of parameters like the area, you see that the area of mitochondria is much more increased in patients. It means that it is a swelling. The swelling of mitochondria means that you lose the capability to maintain a high membrane potential, and the organelle is going nuts, as we say, is going in bad condition. And all the other parameters show the morphological parameters show the same data, and also the functional parameters show that many cells had mitochondria with low membrane potential, and this was done with the methodologies one, a probe which is able to measure the capability of mitochondria to maintain a membrane potential after a stimulus. Then what happened, if you take a look to the in vitro cytokine production, it means that you see that monocytes work properly, even if they tend to produce a little bit more TNF alpha, but not in a statistically significant way. So the meaning is that monocytes have mitochondria which are dysfunctional, but in vitro they are able to produce normal amount of cytokine, which is probably a good news for patients. So jumping back to the conclusions, what we have found that the virus SARS-CoV-2 creates, in my opinion, creates a very big confusion in the immune system, because we have found the presence of T cells with markers of exhaustion, activation, senescence. We saw alteration in the differentiation of different T cell types. We found high plasma levels of a variety of cytokines, those that are pro-inflammatory and those that are anti-inflammatory, and especially those that are related to the Th1 and Th17 phenotype. We found a massive in vitro production of a number of cytokines and the potential skewing towards an Th17 phenotype that we have to investigate much better. We found dramatic differences in B cell population, and we found changes in monocyte population functionality and mitochondrial performance. Before closing, let me acknowledge people who have been working with us in this period. First of all, the group of the infectious disease clinics, because they have seen the very first patient and they were running all the clinical activities in the hospital first, and then even outside of the hospital in the whole area of the Modena province, Cristina Mussini, and who is also my wife, by the way. So we work together in a lot of circumstances, and my life is very difficult, I have to tell you, because she is the director of the clinic and I am the director of the pathology. But we have, in the middle, there is Via Campi, which is a street. So we have a street between the hospital and my lab, which helps. Then I have to thank Massimo Girardis, who is a professor of anesthesia and intensive care, Lucio, Enrico, and all the other guys of the internal medicine, Mario for the help with the viral part, Pia Crismore and Tom Schumacher, who made a lot of experiments on T cell recognition of viral peptides, and finally a group of friends in fluidizing, because we have performed a huge amount of studies with the CYTOKINE technology, which I am not going to present because of the time. And finally, these are the eyes of my group: Sara, Lara, Rebecca, Caterina, Alicia, Lucia, Domenica, Marca, and Maria. They have been working with me without one single day of stop from the end of February to the end of May. And finally, I want to thank you for your attention with the picture that I took in Plaza some time a few years ago. Thank you for your attention.
Thank you, Dr. Cossarizza. It was a very interesting talk. So we are now ready to receive questions. Remember, just to the audience, remember you can write your questions in the chat of YouTube. So in the meantime, I will start with one question. So you showed that CD8 T cells from patients, they produce IL-17.
Yeah.
So Do you know what triggers the production of this cytokine in CD8 T cells?
Do I know what?
What triggers the production? What is the stimulus for the production?
Okay. Well, in this case, also because of lack of time and lack of reagents and lack of cells, essentially, we use the anti-CD3 plus anti-CD28. So most physiological stimulus that we can. So triggering the T cell receptor. Probably it is not the best one, but actually we have, I had no time to show you that we have also used the SAB, Staphylococcal enterotoxin B, a super antigen, and the results were comparable with those with the T cell receptor stimulation. I mean, anti-CD3 plus CD28 or SAB gave more or less the same results.
Mm-hmm. Okay, so here's the question. So it says, regarding the importance of IL-17 in several types of pneumonia, do you still think that it's a good target to block, especially at late-term times?
Absolutely, yes. Points? Absolutely, yes. And in fact, not only we wrote in the paper, in Nature Communication paper, that this was a target, this is a target, but to my knowledge, I am not allowed to tell you some details, but to my knowledge, there is a very big trial in France with a number of different arms. One arm is with secukinumab, that is an antibody blocking IL-17, which is used in therapy. It is something that people use, I guess, in rheumatoid arthritis as well. And the trial is giving very positive results, to my knowledge. But this will come out in a few months, I guess. But I think that it is really a target. And to tell you the truth, in April, we submitted a proposal to the Italian regulatory agency for drugs to start a trial with IL-17, but we were not allowed to do this because they wanted to have a randomized double-blind trial, and we don't want to randomize people because we like people here in Modena. And if a drug works, there is no reason to put someone on placebo. So there was a discussion about that. And in fact, our paper went in the Lancet Rheumatology because the New England did not accept a non-randomized controlled trial. So this is something that you have to consider. I mean, I'm a doctor, I'm a medical doctor, so my interest is to save lives more than to have a paper in the New England. So the decision was to do this because it was very clear that the tocilizumab was working very well. So there was no reason to have a placebo trial. Mm-hmm. They have done that in the UK, but they are different. And I'm happy not to be in the UK.
Okay, so the next question is the cytokine storm the cause or the consequence of the worsening of the patients?
I think the cause of worsening. My impression is that you have a first reaction of the immune system to the virus, but if the first reaction is too high, is too strong, you have troubles. If the first reaction is mild, you're okay. I mean, you have pneumonia, but not with all the consequences of a massive activation of neutrophils that you have there, and a massive activation of endothelial cells. And and there is a paper now that came out very nice in somewhere concerning the autopsies and the molecular and the cellular aspect, the histopathology, and it is very clear that you have a massive activation of neutrophils and monocytes and so on that cause damages, coagulation, thrombosis, and and all you can think about.
Okay, thank you. So there's another question is, what are the parameters that you employ to decide when to initiate a tocilizumab treatment?
Right. So the parameters are not laboratory parameters, are clinical parameters. You have to see the P on F, the P over F, which is the ratio of the pressure of oxygen you have in the blood and the amount of oxygen you use, and your respiratory parameters. You use this, and when you see that it is going down, when you see that you go below maybe 300, and you have to consider also the time in which it is happening. So consider the fall in the respiratory function and the time in which you see the fall, and you can start treating patient around probably 250, 300 to 250. PNEUM, this is a clinical parameter.
Okay.
But you'll see, I mean.
Okay, thank you. So there's another question is, you showed that IgM plasma bloods were increased in COVID patients. What happens with IgG plus plasma blasts?
Well, all plasma blasts were increased in patients. So what we found was that both population of IgM positive or negative were really increasing in these patients. And the idea is that the reason is due to the role of IL-6. You know, IL-6 is a very strong cytokine for B cells. So my impression is that you see that the bone marrow is very much affected by IL-6, and in fact you can check. Well, this is another story. A lot of immature cells are present in the periphery, and you see not only plasma blasts, but immature B cells, I would say, but also immature monocytes and immature neutrophils. So my guess is that a molecule like IL-6 is really doing something terrible. And in fact, blocking IL-6 is a good thing.
Mm-hmm. Okay, another question is regarding the galectin-9, the results that you showed. So it is very high and it is not overlapping in COVID patients versus control ones.
Yeah.
So the question is, do you think that it's a good candidate to be a biomarker for prognostics?
We have to try. We have to do that. We have to try. Yeah, I think it could be. Now, what we are, you know, in that moment where we wrote the paper and signed the paper, and we're still working on that, we were a little bit panicking because we had maybe, I don't know how many people every day to study, to follow, and so on. So we had little time to perform more sophisticated analysis like we are planning to do now, in which we have to put names on the dots. I mean, we have to see what was happening at the different patient level, and to see if a patient who had high level of galectin was bad or not. And we have now we have all the data, and actually we have other data on, if I remember well, 320 patients, which were analyzed for 62 cytokines. So we have a little bit of the data, maybe 20,000 numbers to put together along with the clinical parameters, because we have all the clinical parameters, all the laboratory parameters from the first moment of the entrance to the hospital. So we have a huge database and huge numbers, and it takes time.
Yes, I know.
It takes a lot of time.
Yeah. There's another question is there any evidence of a positive or negative role of innate lymphoid cells in the pathology of COVID-19?
We have some data, yeah. We have some data, I think so. Yes, I think so. I think that they are involved in the pathogenesis in terms of the hyperactivation of these cells. But these are, we have data with CYTOKINE that I am still thinking of what to do with this data. We have some information, but not yet ready.
Okay. Well, I think that we are done. Thank you very much for your excellent talk and for answering our questions. And I hope that next time, yes, you can be giving a seminar in our society here in Buenos Aires that you can come here.
I hope so. I hope so. Okay, thank you to everybody. Thanks to Emilia and to Matthias for the help. And it was a real pleasure to speak with you, even if from Italy. Next time we meet in Buenos Aires.
Okay, thank you very much.
Thank you.
Thank you. Thank you very much, Dr. Cossarizza, for your presentation, excellent presentation. We have many thanks from the audience in the YouTube chat. And I want to thank you, especially because when we invited you to the to the course, to the cytometry course. You could not participate in this moment, but you accepted to give this seminar to us. Thank you so much, and we hope that we can meet you in Buenos Aires in other opportunities. Thank you very much.
I hope so as well. It is a pleasure, I mean.
Okay, thank you. And I will switch to Spanish to finish the webinar. Quiero, en nombre de la Sociedad Argentina de Inmunología, quiero agradecerles a todos de haber asistido al seminario. Creo que es probablemente la última actividad que llevemos a cabo este año. No lo podemos asegurar, pero bueno, es muy probable que así sea. Así que en este caso quiero aprovechar la oportunidad para agradecerles a todos y todas por haber participado y haber mostrado tanto interés en las propuestas que realizamos desde la Sociedad Argentina de Inmunología durante todo este año que ha sido tan difícil para todos. Y quiero aprovechar también para desearles un buen comienzo de un 2021, que esperemos que gracias a las vacunas y a las nuevas terapias que se puedan implementar nos permitan retornar a nuestra vida tal como la conocíamos antes de esta pandemia. Muchas gracias a todos, buenas tardes a todos y todas, nos vemos en otra oportunidad. Gracias.